Okay, very good morning to all of you. Today we are starting our biology lecture of the semester 3, and I congratulate all of you as you are selected for the biology special at our department. So here, as please go through the syllabus, but today's I am just taking the introductory lecture here for our biology special paper. So already we are knowing bryons means the moss and the phyton means the plants. Generally the embryophytes like liverworts, hornworts and the mosses, these are included under this. So what is the embryo? Already we are going to see the life cycle, but after fusion of the male and female gametes, the zygote develop, and this zygote develop into the embryo, which later on gives to the development of sporogenous tissue, right? Or generally as it is called as the development of spores are there. So these spores germinate and again gametophytic plant develop. So here these small plants, they are generally growing in the shady, damp soil areas, and vascular tissue is generally absent, and they reproduce by means of the spores. Uh, Classification of bryophyte is done by a number of scientists, but generally they are divided into three classes as Hepaticopsida, liverworts, Anthocerotopsida, hornworts, and Bryopsida, mosses. Hepaticopsida liverworts divided into four orders as Markane shells. Good example of Markane shells, it is the Rixia and the Markensia, Spirocarpel, Spirocarpos, then Calobrium, it is the one specific genus. Then others are there here as these are the main orders, and the Bryopsida, the different mosses are there. So in Hepaticopsida, mainly these three orders are reported. Here you are observing the good photographs of these Markensia and Archegoniophore, Anthidiophore, then Conosepalum genus on the right hand upper corner, then Gemacopsin Markensia, Rixia flutans at the lower side. Right, so label is about to the photograph. So please see this one. So Gemacops and the Sporogonium, then Rixiocarpus in one specific genus. I think nowadays it is impossible because Markensia found at the high altitude, and we have to go that time for the collection. You see the Corsinia sporophytic or sporogonium nature is there. So these are the some types. Then Pelia is the one genus, it is the larger one genus. You see the thallus, then about the spores and the elaters are there. So in some members Hepaticopsida, the elaters and the spores are present. So just we have to remember this one. Just I am highlighting the characters of these bryophytes. In Anthocerotales, these are good examples of Anthoceros 6 genera: Folioceros, Pioceros, Megaceros, Dendroceros, and Notothylus. So these are our photographs when we have visited to Purandar. Kas plateau is there. Then you see the Anthoceros involucre is there. So it is for the Anthocerotales. In Bryopsida, it is divided into 5 classes: bryals, andrials, spagnels, polytrichales, and buxbaumials. So one part is there, you have to remember the proper names of this one. Then in mosses, the gametophyte is differentiated into protonema and foliose gametophore. Foliose means leafy. Whatever we are observing the moss, it is a foliose gametophore, and which later on developed the antheridia and archegonia and the capsule or the sporogonium. In foliose, they develop axis and leaves without midrib in the sphagnum like mosses, then other mosses showing the presence of mildew. Elaters are absent generally in this one, and rhizoids are multicellular as compared to the anthocerote and the Marcan shells. In anthocerote only smooth wall and the tuberculate, and in Marcan shells they shows the tuberculate and the smooth wall. Then sporogenous tissue develop from the endothecium. Then columella is absent except anthoceros, and some mosses columella is present. Right, we'll see the in detail at the time of the study of each division. Right, then dehiscence of capsule, it is takes place by due to the separation of lead in the mosses. Then in evolutionary character we are observing this. So sporophytic evolution, the steps in evolution they are showing from multicellular sporophyte. The meiosis must be delayed in the zygote, and the zygote must undergo mitotic division to create the multicellular body, that is embryo. Then value of first sporophyte which were nurtured by the gametophyte. Nurtured, that is the they are the totally helping in the development in view of the nutrition. Then gametophyte evolution, it shows multicellular gametangia, that is antheridia archegonia. The sperm cells, that is male gamete, which are developed in the globular club-shaped sacs called antheridia, and eggs are produced in the archegonia. Right. So all these characters we are doing. You see the this flow sheet of the diagrammatic or the mosses are there. So plant body is there. It is generally diploid plant body. Then it shows the spores development in the capsule. So this capsule become, these are the haploid. They shows the vegetative reproduction by means of the gene or by means of this one. And secondly there are the after spore germination, they develop into the male gametophyte or female gametophyte, and separately, and there is the development of sperm. Then the fusion is there, fertilization. So zygote develop into the embryo, and again the spores germinate due to the essence of the sporogonia. So all these, it is all about the introduction of this bryophyte. So thanks to all. We will see the next part in the next lecture.